All right, ladies and gentlemen, so welcome back. So today we're going to pick up our discussion on genetics and really start talking about some additional ways that traits can be inherited. Up to this point, we've been talking really about Mendel and the kinds of traits that he saw in his pea plants, in which case you either had this option or this option. White flowers, purple flowers, tall versus short, wrinkled seeds versus round seeds. But here's the reality. A lot of characteristics in organisms aren't inherited that way. In fact, Mendel was incredibly lucky or maybe just smart in the traits that he happened to choose in studying those pea plants in the first place to be able to understand the inheritance patterns that he discovered. When we look at inheritance patterns of a lot of other characteristics, they often will fall into one of these four categories instead of that traditional Mendelian inheritance. So intermediate inheritance, codominance, polygenic inheritance, and sex-linked traits are the ones that we're going to focus on for this year. That being said, there are other options even above and beyond this, but for our scope, let's keep it just to these four. So let's pick up today and talk about intermediate inheritance first. You may come across this also known as incomplete dominance, so just know that either of those terms are potentially referring to the same thing. And what we're talking about here with intermediate inheritance or incomplete dominance is a situation where you have sort of a quote-unquote dominant allele, but it's not truly able to fully mask the expression of the recessive one. So when we talk about Mendelian genetics and that dominant versus recessive, think of it maybe in percentages if that helps. When you have a heterozygote individual, their dominant characteristic 100% masks the recessive protein that's being produced. So although they've got both a dominant and recessive allele, they're producing two separate proteins. And when we see an individual who's heterozygous, that dominant protein 100% masks the fact that that recessive protein still exists in the cell. But when we get to a situation with intermediate inheritance or incomplete dominance, that dominant allele isn't able to 100% mask the recessive one. So when I look at a heterozygote individual, all right, and you observe the phenotype that gets produced, that quote unquote dominant allele maybe is only being able to mask the recessive one, say 50%. And so as a result, you end up with an intermediate result. You end up with, instead of two distinct phenotypes from three genotypes, you now have three genotypes with their own individual unique phenotypes. So 3 genotypes to 3 phenotypes when we look at intermediate inheritance. That being said, be careful because when we look at some of the examples of these, it's going to look like you're kind of blending paint colors together. I'll show you an example with flowers in just a second and you'll see what I mean. But keep in mind that intermediate inheritance is not the same thing as that blending hypothesis that we talked about way back at the beginning of this section. When we talk about blending hypothesis, it was literally like blending paints together, taking red plus white and making pink. The difference here is, though, with that blending hypothesis, it is like paint. There's really no way to get that red paint and that white paint back out. The reason I say that this is not like blending is because remember that Mendel did ultimately end up supporting that particulate hypothesis instead, where I've got those individual alleles being passed on as discrete particles, in which case they can come back in that F2 generation. If I take that parent generation where they're both homozygous for their trait, put them together in that F1 where I get all those heterozygotes, mate those heterozygotes together. Remember in that F2 generation, those parental traits came back. So it's still going to follow that traditional 1 to 2 to 1 ratio when I look at intermediate inheritance, that we saw that same 1 to 2 to 1 genotypic ratio back in Mendel's traits as well. So I'm still going to get one individual who's homozygous dominant. I'm going to get two individuals who are that traditional heterozygote, and I'm going to get one individual who's that traditional recessive. The only difference here is when we talked about Mendel's traits, it was 1 to 2 to 1 genotypic, and then 3 to 1, 3 dominant to 1 recessive in your phenotypic ratio. When I look at intermediate inheritance, I still get that 1 to 2 to 1 genotypic, but now it is also my phenotypic ratio. So that's a big difference here. So if I look at some examples of intermediate inheritance, here's one with betta fish. So green is not completely dominant over that royal blue color. So when we write these out, we typically use 2 capital letters. And so here you'll get 2 G's. That gets me that sort of traditional dominant green. I get 2 B's, more your traditional dominant, or excuse me, more your traditional recessive. But when I get both of them, I get a mixture of this green and blue, and so I sort of get this sort of steel blue color in the interim. So instead of being all dominant and then a recessive, now I've got 3 distinct phenotypes for the 3 different mixtures of genotypes that I can have. Here's another example with the flower color. So if I take that parental generation where I have a purebred red, right? So I see these 2 R's, purebred white, 2 W's, mate them together, I get that F1 generation. Here's what you would typically think of as that heterozygote. I've got 2 of each, and I get now a distinct third phenotype in the color pink. But why is this not that blending hypothesis? Because I go down to that F2 generation where I mate these 2 guys together, and now I get the red and the white back. If this were truly blending, they'd still remain pink. So that's the big distinction here. All right, moving on to another example of non-Mendelian inheritance is codominance. So here, none of the alleles are truly dominant or recessive. If I have that normal, what you would think of as heterozygote individual who has both alleles, now both alleles are equally as strong to each other. So there is no masking at all. So go back to what I was just saying. We take Mendel's dominant and recessive, and that dominant 100% masks the recessive. If I look at intermediate inheritance, it's maybe a 50% masking. Now when I get to codominance, there's no mask at all. Both alleles are equally as strong, and so we talk about them as being codominant to each other. So when both alleles are present, they are both expressed. So how do we write this as a way to sort of differentiate? Well, up to this point, we've been doing capital versus lowercase for Mendelian genetics. I just showed you the 2 different capital letters for intermediate inheritance. In codominance, we're going to use a single letter and then use 2 different capital letters as superscripts. All right. So if I show you what that looks like here with these Andalusian chickens, so they've taken the C's and then they've used B or W as that superscript. So if I take that parent generation where they're purebred for black, there are my 2 capital B's, purebred white, all right. And then my intermediate inheritance, or excuse me, my codominant, the C and the B and the W, they are these guys right here. Now, don't kind of confuse this with intermediate inheritance because I know it can kind of look like that from the picture. What you would do is when you zoomed in on that particular F1 chicken, you'd actually be able to see individual black and white feathers, so they would be separated. You pull out, look at the chicken as a whole, and that's sort of where this bluish grayish color comes from, but zoom in and you would be able to see individual white and individual black feathers. Go ahead and mate 2 of those guys together to make that F2 generation, and I'm still seeing that 1 to 2 to 1 phenotypic ratio. It's just now when I look at those heterozygote individuals, these guys here, both traits are equally as strong. There's no masking going on at all. Another great example of codominance is your human blood typing. So let's take a second and really talk about how blood typing works. So when we talk about your blood type, we're talking about antigens or these surface markers on your cells. Specifically with blood typing, they're on your red blood cells. And so if I look at an individual who is type A, they create these A surface antigens on their red blood cells. If I look at somebody who's type B, they create a different antigen, that B antigen, on their red blood cells. Here's what we were talking about with that codominance. So they create both A and B antigens because they now have both alleles. Now, blood typing is also an example of not only codominance, but an example of multiple alleles, so more than two different options. And in your ABO system for blood typing, you have three options. You've got an A, a B, and an O. So if I have the A and the B, I now show both. If I have B only, then I show B. If I have A only, I show A. And in the case of having O only, I don't actually produce any antigens at all. So this is where your blood type comes from. It's by looking at the antigens, and all of this is genetically controlled. Why does this matter? Well, it comes down to blood transfusions and tissue matching when we talk about organ transplants. It's really important because we can't cross type. What does that mean? I can't give you a blood type that doesn't work with your own body system. And the reason really for that has less to do with the antigens themselves and more to do with these antibodies that your body produces against the opposite antigen. So antibodies are proteins that your white blood cells create, and they will create them against things that are not yourself. So these antibodies, in the sense of your blood type, they produce the antibodies that are opposite of the antigen that you possess. So if I look at somebody who is type A, they've got those A antigens, and so their antibodies are anti-B. They are the opposite shape for the opposite antigen that my cells don't have. I go to type B, now I'm producing the B antigens on my blood cells, and so my body produces anti-A against that opposite antigen. If I'm type AB, I can't produce either antibody, and the reason for that is because anytime that antibody comes in contact with its antigen, they connect. It's like fitting puzzle pieces together, or think of it like the enzyme substrate connection. They're fitting together. And anytime that antibody finds its corresponding antigen, they stick and they clump together. Well, if I gave you the wrong blood type, and now you get a donor's blood that has the opposite antigen, your antibodies are going to stick to that antigen on the surface of that red blood cell and cause the blood that I've just now given you, presumably because you're in some dire need for it, because of injury, surgery, whatever the case might be. I'm now worsening the situation because I'm now causing that fresh blood I've just given you to coagulate, to agglutinate is the term for it, to clot up in your bloodstream, and that's just not what we want. That's a bad situation to be in, and often, historically at least, would result in death. So your person who is AB, your sort of co-dominant individual, can't produce either antibody because they would stick to their own red blood cells and cause their blood to clot. If I look at somebody who's type O, because they have no antigens, they actually produce both. So I've sort of alluded to this idea of why does this matter? Well, it really matters because of those blood transfusions. So when you go back to, say, the 1800s, early 1900s, and look at these first blood transfusions, we didn't understand that this typing was a thing. We didn't understand that antigens and antibodies existed. And so more often than not, in our effort to save people and give them blood, we killed them in the process because we were giving them the wrong blood type and causing the blood that we just gave to them in that life-saving measure to clump up and stick. So when we look at this, all right, if I'm type O, if I'm type A, that means I have A antigens on my red blood cells, so my body produces anti-B antigens. So what does that ultimately mean then in terms of who I can and cannot get blood from and who I can donate to? So type A, I can get blood from myself or from anybody who has the same blood type as me. So I can get type A. I can also get from type O, because remember, type O red blood cells have no antigens, so I'm good. I don't have to worry about my anti-B antibodies connecting to anything because there's nothing to connect to. In terms of donating, I can donate to anybody who's like me. I can also donate to anybody who is AB because they have no corresponding antibodies. So we're good and clear there. Type B would be the opposite. So I've got B antigens, anti-A antibodies, so I can get blood from anybody who's type B or type O, and I can give to anybody who's B or type A. If I look down at type AB, well, now I've got both antigens, but no antibodies, which means I can get from anybody because I don't have any antibodies to stick to any sort of foreign antigens. I'm good, I'm clear. That being said, I can only donate to somebody who is AB, and O is the opposite of that. So they've got no antigens, but because they've got both antibodies, they can only get from type O, but they are able to donate to anybody. So when you talk about blood transfusions and you hear the terms universal donor versus universal receiver, we're really talking down there about type A and type O. So type AB would be your universal receiver. They can take from anybody, they're all good. Type O is your universal donor where they're able to give to everybody. And it really comes down to whether or not those antibodies are present. When we talk about it from a genetic standpoint and look at their genotypes versus phenotypes, there's some unique situations that get set up here. Not only is this a situation of codominance, but it's also a situation of having more than just two alleles, which is really all that we've been dealing with up to this point. So there are multiple alleles involved. In fact, there's 3 of them. So if I look at somebody who is type A, right, so they can be what we would traditionally think of as our homozygous recessive, or excuse me, are homozygous, just homozygous in general, where they are 2 capital A's, but they can also be an interesting heterozygote mix where they can be AO as well. So type A can be 2 A's or an AO. Type B could be 2 B's or a BO. Type AB has to be AB, that's your codominant, and then type O is recessive to everything, and so type O only has the double O. Another type of unique inheritance pattern we can discuss is polygenic inheritance. And so up to this point, we've often utilized things like eye color, skin color as examples of traits, but we've sort of been oversimplifying it and assuming that it's either blue eyes or brown eyes, brown hair or blonde hair, and there really isn't that sort of cut and dry situation with a lot of those characteristics. Instead, they're polygenic. So remember, poly means many. So in the case of polygenic inheritance, I'm talking about a single trait, the eye color, the skin color, the height, for example. But rather than it being controlled by a single location in your chromosome, so a single gene, and you either inherit 2 of one copy or you're heterozygote. In this case now, I'm talking about a single trait that is being controlled by multiple genes, either on the same or on different chromosomes. So I now have a range of characteristics to fit within that. So think of skin color, for example. You look across the globe and there is a wide range of skin color. Why? Because it's polygenic. You're not just talking about this or that. You've got multiple options based on how many dominants versus how many recessives you inherit. So here's what I'm talking about, right? So if I assume skin color is taken care of by 3 different genes on 3 different locations in my genome, then the more recessives that I have, the lighter I am, the more dominance I am, the darker I am, and most of the population falls somewhere in between by inheriting some combination of dominance and recessives. Eye color works the same way. All right, so there are multiple alleles that control those, and that's why we get that range in coloration in your eyes as well. And fur color, same thing in something like our cute little puppies here. All right, there's a range of coloration. Why? Because there's more than one characteristic, or more than one gene, I should say, controlling that characteristic in your genome. One sort of offshoot that's kind of an interesting idea wasn't necessarily in that initial list that I gave you, but it's an interesting one to talk about. That's also deviates from Mendel's traditional laws is seen in these cats. And if you look close, do you notice anything a little weird? Well, hopefully you recognize they have no tail. This isn't because it's some trick of Photoshop. Minx cats are actually a breed that is truly born without a tail. And when we look at the way their traits are inherited, it initially appears like traditional Mendelian genetics. To be tailless is dominant, and to have a tail is recessive in this particular breed. So as a breeder, you're thinking, cool, all right, it should be really easy to breed a bunch of tailless cats and always get tailless cats, right? If I constantly breed that dominant together, I should be able to create a pure homozygous dominant line and then continue to always get tailless cats from there. Here's the catch though. If I take two Minx cats and I mate them together, I will always get, statistically speaking, two tailless kittens for every one tailed kitten. Now, do the math here a second. I said this looks very much like traditional Mendelian genetics. Think about your Punnett squares. You should have 4 offspring in a cross, correct? 4 boxes in your Punnett square. And yet, do the math here real quick. I've got 2 tailless kittens for every 1 tailed kitten. 2 plus 1 equals 3, not 4. And so here's where this does end up deviating from Mendel's initial expectations. And the reason for it is that to be homozygous dominant for this genotype is what we call embryonic lethal. So what does that mean? It causes the embryo to fail to develop. So any individual embryo that would have inherited 2 dominant Ts, 2 tailless genes from each parent, never actually gets born. And so every Minx cat therefore has to be heterozygous for its genotype, which means as a breeder, you will always end up with the potential to get a handful of tailed kittens in your breed, which does also mean that this particular breed is a little bit more expensive. From a breeder standpoint, you got to figure out what to do with these tailed kittens because it's not what you're trying to create. It's not what your clientele is looking for. So when we look at a true heterozygote cross, 2 actual living Minx cats have to be heterozygous. So when I cross them together, any individual cats that end up with that homozygous dominant are lethal. They never persist, they never become actually living, functioning kittens, but you will get 2 Minx cats for every tailed cat that you do possess. So for some reason, something in having that double copy of that tailless gene and the way that that develops proteins or signaling or whatever the case might be in that offspring ends up failing that embryo from developing.